Welcome to the Deep Dive. Our mission today, well, it's ambitious. We're aiming to go way beyond the basics and achieve, really, a professional mastery of the body's internal architecture.
That's right. We're talking about the circulatory and lymphatic systems.
The whole machinery of flow, and we're going deep from the molecular level right up to the major systems.
Exactly. So for those of you really aiming for that professional level grasp, we're bridging the gap from the microscope right up to the macro view.
Okay.
The goal is to get a deep understanding of the two key muscle types driving all this internal fluid movement. You've got cardiac muscle,
The big pump in the heart.
the ultimate centralized pump, yeah. And then there's smooth muscle. This is the masterful, really plastic regulator of pretty much every vessel diameter and hollow tube in the body.
So it's not just about what they do.
No, not at all. We need to get into the surprising cellular mechanics that actually allow them to be so adaptable. That's where the real insights are.
Right. Because when most people think muscle, they picture skeletal muscle, the voluntary stuff that moves our arms and legs.
The striated kind.
Exactly. But the systems keeping us alive, they rely on the involuntary world. So our deep dive really has to start with the most widespread regulator in that world, smooth muscle.
Makes sense. It controls all blood flow once you leave the heart.
And its identity is basically defined by what it's not, right? It's involuntary, no conscious control, and unstriated.
Meaning those contractile proteins, the actin and myosin, they aren't lined up in those neat, regular sarcomeres you see in skeletal or cardiac muscle.
And that lack of strict organization is actually its strength.
Absolutely. It grants smooth muscle this tremendous functional range, this plasticity. It really is the jack of all trades in the muscle world.
Okay, like what kind of roles are we talking about?
Oh, all sorts. Regulating diameter is a big one. Think bronchial tubes in the lungs, all the blood vessels. Then there's propelling contents along hollow organs.
Like the intestines.
Intestines, the ureter carrying urine, the oviduct, and even forcefully expelling contents. Think about the uterus during childbirth or the bladder during urination. That's all smooth muscle power.
Structurally, what jumps out about these cells? You mentioned variability.
The variability is incredible. Yeah, they're typically described as spindle-shaped, sort of tapered at the ends. But the scale, I mean, it's unbelievable. You might find tiny ones, maybe 15 micrometers long, in a small blood vessel wall. But then in a pregnant uterus, they can stretch to over 500 micrometers, half a millimeter long.
Wow. That's a huge range.
Oh.
And it must reflect their function.
It absolutely reflects the required function,
Wow.
critically. And visually, if you're looking under a microscope, say, as a pathologist, you're looking for a single nucleus, usually right in the center of the cell.
Okay.
And here's a really neat functional marker. When that cell contracts hard, the nucleus often twists up. It contorts into this distinct corkscrew shape.
A corkscrew.
Yeah. Seeing that corkscrew morphology is a high-value diagnostic sign. It tells you immediately, okay, that smooth muscle cell is contracted.
So you have these individual tapered cells varying wildly in size. How do they manage to pack together and contract something large like the gut wall without the force just, fizzling out?
That relies on some pretty sophisticated structural planning and specialized connections between the cells.
How so?
Well, first, the cells are staggered longitudinally. So the wide middle part of one cell sits next to the tapered ends of its neighbors. This creates incredibly close packing.
For efficient force transfer.
Exactly.
Yeah.
And if you slice a bundle of smooth muscle and look at the cross-section, you see these profiles of different sizes. But the nuclei, they only show up in the biggest profiles right in the very center of the cells. It's quite different from skeletal muscle, where the nuclei are pushed to the edge.
Okay, but they aren't anchored by big tendons like skeletal muscle. So what's physically holding the whole contracting unit, the fascicle, together?
Right, no tendons here. The secret is really the extracellular matrix bridge.
Extracellular matrix.
Yeah.
The stuff between the cells.
Yeah. Smooth muscle cells are separated by this small gap, maybe 40 to 80 nanometers wide. Each cell wraps itself in a kind of coating called a basal lamina.
Okay.
And this lamina merges with a layer of connective tissue around it, a reticular layer made of fine collagen, types I and III mostly, and some elastin. This material literally bridges the gap between cells.
So it provides mechanical continuity.
Precisely. When one cell pulls, its neighbors feel it directly through this matrix connection. The whole fascicle moves together.
Now, you mentioned something earlier that sounds incredibly significant, especially thinking about diseases.
What's that?
You said the smooth muscle cells make their own matrix.
Yes. That's one of the most critical insights. Unlike most tissues where you have specialized cells like fibroblasts churning out the collagen and elastin,
The smooth muscle cells do it themselves.
they synthesize their own matrix components, the collagen, the elastin, the ground substance, all of it.
And that defines their plasticity.
It's absolutely central to their plasticity. It allows them to actively remodel their surroundings, in response to changes in load or injury. This is fundamental to understanding vascular diseases like hypertension or arteriosclerosis, where the vessel walls change pathologically.
How do they anchor themselves within this matrix they've built?
They use specialized spots on their membrane called submembraneous dense plaques. These contain proteins called integrins that grip onto the matrix, and they connect to their neighboring cells using structures called intermediate junctions. They're kind of like small versions of desmosomes you see elsewhere.
Okay, let's dive inside the cell now. The contractile machinery. It's not a neat sarcomere, you said? So what does this lattice look like? How does it allow for that massive shortening?
Right, it's quite different. The cytoplasm, basically everything outside the area immediately around the nucleus, has two main components structurally.
Okay.
You've got the cytoskeleton, which gives the cell its shape. That's mainly built from intermediate filaments, things like desmin, and vascular muscle, vimentin, too.
And where do these filaments attach?
They insert into structures called cytoplasmic dense bodies within the cell, and also into those dense plaques we just mentioned under the membrane. Think of these dense bodies and plaques as the functional equivalent of Z-discs in striated muscle. They're the anchoring points.
Got it. So those are the fixed points. Where's the actual power generated?
The power comes from the contractile apparatus. This is a complex lattice, a network, really, of actin and myosin filaments. But here's the key. They're arranged obliquely, kind of crisscrossing throughout the cytoplasm, not in straight lines like sarcomeres.
Obliquely.
Yeah.
So diagonal.
Sort of, yeah. This oblique lattice structure is what allows the force generated by actin and myosin sliding to be transmitted out to the plasma membrane via those dense plaques. And crucially, the molecules themselves are different from striated muscle.
Okay, lay out the molecular differences for us. What makes them so flexible?
Well, first, the ratio of actin to myosin filaments is huge. It's about 8 times more actin relative to myosin compared to striated muscle.
8 times.
Yeah. And the myosin itself is a slower isoform. It hydrolyzes ATP, the energy molecule, more slowly. The myosin filaments are also physically longer, around 1.5 to 2 micrometers.
And you mentioned something about their shape.
Right. Most importantly, they're described as ribbon-like. This means they have the myosin heads, the little motors that grab actin, sticking out symmetrically along the entire length of the filament.
Unlike striated muscle myosin, where heads are only at the ends.
Exactly. This ribbon-like structure means actin filaments can slide along the whole length of the myosin filament. There's no central bare zone.
Ah, okay. So the actin can slide along the entire length. That must be how they achieve such incredible shortening.
That's precisely it. That structural difference explains why smooth muscle can contract by over 80% of its resting length. It's dramatic compared to the roughly 30% limit you get with sarcomere-based contraction.
80%, that's massive.
It is. But there's a second huge advantage, especially for organs that need to maintain constant tone, like arteries holding blood pressure. The low energy requirement. Because that myosin ATPase cycle is so slow, the cell can maintain tension for really long periods without burning through ATP rapidly. This sustained tension state is sometimes called the latch state.
Latch state, so it can kind of lock in the contraction.
Sort of, yeah. It's incredibly efficient for maintaining things like blood pressure all day long without fatigue. Vital function.
Okay, so we have this incredibly adaptable architecture. How does the nervous system manage to control such diverse functions? I mean, controlling the iris in your eye seems very different from the waves of digestion.
Right, the innervation isn't one size fits all. It really exists on a spectrum.
Spectrum.
Yeah. At one end, you have what are called multi-unit smooth muscles. Think about the muscles controlling most blood vessels, or the ciliary body in the eye that focuses your lens.
What defines them?
They typically have dense innervation. Each cell, or small groups of cells, might get its own nerve input. This allows for a really precise local control, and the cells tend to operate more independently of each other.
Okay, so that's one end. What about the systems that need to act as a big coordinated sheet, like the gut?
Those are generally unitary smooth muscles. You find these in the GI tract, the ureter, the uterus. Here, the nerve supply is much sparser.
Sparse innervation? How do they coordinate then?
The nerves often just modulate the baseline activity, These muscles have intrinsic myogenic activity. They can contract spontaneously just on their own or in response to being stretched.
So they don't always need a nerve signal to contract.
Not necessarily to initiate it, no. And the key for coordination is that their cells are extensively linked by gap junctions.
Ah, the electrical connections.
Exactly. These allow the electrical excitation to spread rapidly from cell to cell, so the whole sheet of muscle contracts more or less as a single unit. Global influence.
And the actual signal transmission, the synapse.
Mm-hmm.
It's fuzzier than in skeletal muscle, right? No tight neuromuscular junction.
Yeah, correct. It's much more diffuse. Smooth muscle gets its signals from unmyelinated axons coming from autonomic ganglia. These axons branch out, and along the branches, they have these little swellings called varicosities.
Beaded axons.
Yeah, looks like beads on a string. Each varicosity is a site loaded with neurotransmitter vesicles and mitochondria, ready to release the signal. A single neuron can have tens of thousands of these varicosities.
Huge reach, then.
Enormous reach. But the gap between the varicosity and the muscle cell membrane, the junction, is highly variable.
How variable?
It can be as close as 10 nanometers, pretty tight, or it can be as wide as a full micrometer.
A micrometer, that's a huge gap.
It is. It means the neurotransmitter often has to diffuse quite a distance to reach its receptors. This leads to a slower, more widespread influence across a whole field of cells, which actually suits that unitary pattern of contraction quite well.
Okay, let's transition to the trigger mechanism itself, excitation-contraction coupling, or ECC. You said this is where smooth muscle really diverges.
It really does. The complexity comes partly because there isn't just one way it happens. There are two main coupling mechanisms.
Two.
Yeah. First, you have electromechanical coupling. This is driven by an action potential or just a change in membrane voltage depolarization. It's common in those phasic unitary muscles, and the signal spreads cell to cell via the gap junctions.
Okay, depolarization triggers it. What's the second way?
The second and really critical mechanism, especially in tonic muscles like in blood vessels and airways, is pharmacomechanical coupling.
Pharmacomechanical, meaning it's triggered by chemicals, not just voltage.
Exactly. It doesn't rely on a big change in membrane potential. Instead, it's triggered by signaling molecules, agonists like noradrenaline or endothelin binding to receptors on the cell surface. These are usually G protein-coupled receptors.
And this is the major way vascular tone is controlled.
It's the major mechanism in vascular smooth muscle, yes. And the absolute central regulatory difference compared to striated muscle comes down to calcium.
How so?
In striated muscle, calcium binds to troponin C to initiate contraction. In smooth muscle, troponin C is largely absent. Instead, calcium binds to a different protein called calmodulin.
Calmodulin, okay.
Wow.
So calcium hits calmodulin. What happens next to cause that slow, efficient contraction we talked about?
That calcium-calmodulin complex then activates a key enzyme, myosin light chain kinase, or MLCK.
MLCK.
MLCK is the crucial player. Its job is to phosphorylate, to add a phosphate group to the regulatory light chains on the myosin head.
And that phosphorylation is the on switch.
That's the trigger. Once those light chains are phosphorylated, the myosin head can interact with actin, start the slow ATPase cycle, and generate force. Contraction begins.
So the amount of contraction, the tone, must depend on how active MLCK is.
Precisely. It's all about the dynamic balance between the activity of MLCK, which promotes contraction, and another enzyme called myosin phosphatase.
Which does the opposite.
Exactly. Myosin phosphatase removes that phosphate group from the myosin light chains, promoting relaxation. The level of muscle tone at any moment depends entirely on the relative activity of these two enzymes.
Can this balance be actively manipulated?
Oh, absolutely. It's a major target for regulation. Many vasoconstrictor signals, for instance, don't just increase calcium, they also act to inhibit the myosin phosphatase. They often do this via another signaling pathway involving rokinase.
Wait, so if you inhibit the enzyme that causes relaxation,
you increase the net level of myosin phosphorylation and thus increase force, even without necessarily increasing the calcium levels much more.
that's a huge point. It means the muscle becomes more sensitive to the calcium that's already there.
Precisely. This calcium sensitization is a central mechanism. It allows smooth muscle to generate significant force even at relatively low or resting calcium levels. And unfortunately, it's also a central feature in diseases like hypertension, where you get this pathologically elevated vascular tone.
And where does the calcium itself come from? Is it all from outside the cell?
Not entirely. Intracellular stores in the sarcoplasmic reticulum, the SR, play a big role too. Often, Pharmacomechanical coupling involves the generation of IP3, inositol tresphosphate.
The second messenger.
Right. IP3 triggers calcium release from these internal SR stores. But here's something really interesting, kind of cutting edge.
Yeah.
It might not just be the average calcium level inside the cell that matters most. Some researchers now think that the rate and pattern of slow calcium oscillations, these little waves of calcium release, might be the prime driver for specific smooth muscle functions.
Oscillations, not just a steady level.
Yeah. These oscillations could potentially fine-tune everything from tonic contraction levels to even influencing gene transcription and cell growth or proliferation. It's a really active area of research.
Okay, that detailed look at smooth muscle gives us the molecular toolkit. Now let's zoom out. Let's scale up and look at the whole systemic architecture that these cells control the cardiovascular system itself, mapping the vascular tree.
Right. The scale is just immense. You start centrally with the aorta, maybe 30 millimeters across.
Big pipe.
Huge. Then the arteries branch and branch and branch. They increase massively in number, but each individual vessel gets smaller and smaller in diameter as you move out to the periphery. You end up with arterioles down around, say, 10 micrometers.
So what's the big functional consequence of all that branching? Does it slow things down?
It absolutely slows things down. Even though the individual pipes get tiny, the total cross-sectional area across all those parallel branches increases enormously.
How much?
At the level of the arterioles, the total area might be something like 200 times the area of the aorta.
200 times.
Yeah. So basic physics tells you if you increase the area that much, the velocity of flow has to drop dramatically. And that's crucial. You need slow flow out in the capillaries to allow enough time for oxygen, nutrients, waste products, all that exchange to happen effectively.
And the vessel walls themselves must change fundamentally along this path to reflect different jobs.
Oh, completely.
Yeah.
Especially thinking about conducting versus resistance vessels, the wall thickness gradient is key. Well, the absolute thickness of the arterial wall does decrease as you go towards the periphery. But relative to the size of the lumen, the hole in the middle, the wall actually gets much thicker.
Thicker relative to the opening.
Yeah. In a big elastic artery like the aorta, the wall thickness might only be, say, 1/115th of the vessel's radius. But when you get down to a tiny arteriole that's controlling flow, the muscular wall can be almost half the radius.
Wow. So it has massive leverage.
Immense leverage. That thick muscular wall gives the arteriole incredible power to constrict and control peripheral resistance, directing blood flow.
Okay, that's the outflow tract. What about the return journey? The venous system. You mentioned capacity earlier.
Right. The veins are the system's reservoir. They're the high capacitance part of the circuit. They start small out in the periphery and then converge, joining together into larger and larger vessels as they head back towards the heart, ending in the vena cava.
So the total cross-sectional area decreases again.
It decreases towards the heart, yes. And the key feature of veins is their high compliance. They stretch very easily.
Compliant. Like a floppy balloon versus a stiff pipe.
Kind of, yeah. They don't have the thick muscular or elastic walls of arteries. This high compliance, combined with the fact that pressure is low on the venous side, means they can hold a huge volume of blood. The majority of your blood volume actually is sitting in your veins at any given moment.
Before we dive into classifying the different artery types, you mentioned something about crucial architectural arrangements, neurovascular bundles.
Yes, these are important. You often find large arteries running right alongside their main accompanying veins, often called vena comitants or satellite veins. Nerves usually run with them too, hence neurovascular bundle.
And why is this close association significant?
It allows for countercurrent exchange. Heat is a big one. In your limbs, warm arterial blood flowing out heats up the cooler venous blood returning towards the core. This helps preserve body heat.
So it acts like a heat exchanger.
Exactly. And you see similar principles for solute exchange in other places, like the vasorectal blood vessels looping down into the kidney medulla. That countercurrent exchange there is absolutely essential for concentrating urine.
Okay, let's classify the arteries now. Structurally, most vessel walls, except the very smallest, have 3 layers, right?
That's the standard model. The inner tunica intima, the middle tunica media, and the outer tunica adventitia.
And we classify arteries based mainly on the middle layer, the media.
Primarily, yes. The composition of the media tells you the vessel's main job. So first you have the elastic arteries, also called conducting arteries. Think aorta and its major branches.
Elastic. So lots of elastin.
Dominated by it. Their media isn't mostly muscle, it's packed with elastic tissue. It's organized beautifully into these concentric layers, maybe around 50 or so lamellar units. Each unit is a sheet of fenestrated elastic tissue, alternating with a thin layer of smooth muscle and collagen.
And this structure is all about handling the heart's output.
It's about buffering that pulsatile pressure wave from the heart. During systole, when the heart ejects blood, these elastic walls stretch, absorbing the pressure shock.
Like a shock absorber.
Exactly. Then, during diastole, when the heart relaxes, the stretched elastic walls recoil. That recoil pushes the blood forward, maintaining continuous flow downstream and preventing the pressure from dropping drastically between heartbeats. Smooths out the flow.
Okay, so elastic arteries conduct and smooth the flow. What comes next?
Next are the muscular arteries, or distributing arteries. These are most of the named arteries you learn in anatomy, like the brachial or femoral artery.
Muscular, some more muscle here.
Much more muscle. Here, smooth muscle really predominates. It can make up 75% or even more of the media's mass. The muscle fibers are typically arranged spirally or circumferentially around the vessel.
And their job is distribution.
Right. Their contraction and relaxation controls where blood is directed within the body, shunting it to different organs or regions based on need. Structurally, a key feature is a very prominent, often wavy-looking internal elastic lamina separating the intima from the media.
Wavy because the muscle is contracted.
Often, yeah, when the muscle contracts, it kind of bunches up that elastic layer.
And the final control point. The most important resistance vessel. You mentioned arterioles earlier.
That's the arteriole. These are the tiny workhorses. They might only have 1 or 2 layers of smooth muscle cells wrapped around them.
Doesn't sound like much.
But remember that relative wall thickness. Compared to their tiny lumen, that muscle layer is incredibly thick and powerful. Functionally, arterioles act as precapillary sphincters.
Like little taps or floodgates.
Exactly like taps. They are the primary site where peripheral resistance is regulated. By constricting or dilating, they control how much blood flows into the downstream capillary beds in any given tissue. And they're very sensitive, densely innervated, especially by sympathetic adrenergic nerve fibers telling them to constrict.
Now, sometimes the circulation takes a detour, right? These portal systems.
Ah, yes. Fascinating exceptions to the standard artery capillary vein sequence. They involve 2 capillary beds connected in series.
2 capillary beds. How does that work?
Well, in venous portal systems, you have a setup where blood flows from one capillary bed through a portal vein directly into a second capillary bed before finally returning to the heart.
Where do we see that?
The most famous example is the hepatic portal system. Capillaries in your intestines absorb nutrients, collect into the hepatic portal vein, which then breaks up into another capillary network, the liver sinusoids. This delivers nutrient-rich blood directly to the liver for processing.
Smart design.
Any others? The hypothalamo-hypophyseal portal system is another critical one, linking the hypothalamus to the anterior pituitary gland for hormone transport.
And is there an arterial equivalent?
There is. An arterial portal system has a capillary bed sandwiched between 2 arteries, or more precisely, between an arteriole and another arteriole.
Where's that found?
The classic example is in the kidney, the renal glomerulus. Blood flows in through the afferent arteriole into the glomerular capillaries where filtration happens, and then flows out through the efferent arteriole.
Why structure it like that?
It's essential for maintaining high pressure within the glomerulus. The efferent arteriole provides significant resistance to outflow, keeping the glomerular capillary pressure high, which is necessary to drive fluid filtration out of the blood.
Okay, we've followed the pipes down to the business end. The microvasculature, the exchange vessels. How are they classified?
It mainly comes down to how leaky their walls are, specifically the endothelial lining. The tightest are the continuous endothelium capillaries.
Continuous meaning no gap.
Essentially, yeah. The endothelial cells form an unbroken lining, joined by tight junctions. Exchange happens mainly via small molecules diffusing across the cells or through tiny intercellular clefts, and also via penocytosis, little vesicles shuttling fluid across.
Where do you need that tight control?
You find them in tissues where you need to regulate passage carefully: muscle, connective tissue, nervous tissue. And they form the basis of the super tight barriers, like the blood-brain barrier or the blood-testis barrier, where extremely tight junctions severely limit even small solute passage.
Okay, what if you need faster exchange?
Then you have fenestrated endothelium. Fenestra means window. These endothelial cells have numerous small pores or fenestrations right through their cytoplasm.
The holes in the cells.
Yeah, about 50 to 100 nanometers across. Often, these pores are covered by a thin diaphragm made of glycoproteins, like one called PV1, but they still dramatically increase permeability to water and small solutes.
Makes sense. Where are these common?
Wherever you need rapid fluid and solute exchange endocrine glands releasing hormones into the blood, the intestinal lining absorbing nutrients, the choroid plexus making cerebrospinal fluid, and importantly, the renal glomeruli for filtration.
And the most permeable type, the leakiest of all.
Those would be the sinusoids, or sinusoidal capillaries. These are wider, more irregularly shaped capillaries than the others, and their walls have true discontinuities.
Discontinuities, like actual gaps.
Yes, actual gaps between the endothelial cells, not just pores within them.
Yep.
Plus, the basement membrane, that basal lamina layer underneath, is often incomplete or totally absent.
So they're really leaky.
Extremely. They're designed for maximum exchange, allowing even large molecules and blood cells to pass easily between the blood and the surrounding tissue parenchyma. You find sinusoids in places like the liver, bone marrow, spleen, and some endocrine organs where this intimate contact is essential.
Okay, blood filters through the capillaries or sinusoids. Where does it collect immediately after?
It converges into the postcapillary venules. These are the smallest veins, maybe 10 to 30 micrometers wide.
Why are these seemingly small vessels so important?
They are incredibly important. Initially, they lack a proper smooth muscle layer. Their key features are high permeability, even more permeable than capillaries sometimes, and they are the primary site for the inflammatory response.
Inflammation central.
Yes. When tissues get injured or infected, it's mainly across the walls of these postcapillary venules that fluid leaks out, causing swelling. And, crucially, where white blood cells, leukocytes, migrate out of the bloodstream into the tissues to fight infection, they are a major part of the microcirculatory exchange bed.
All right, let's zoom in again, right down to the single cell layer lining all these vessels.
Yeah.
The endothelium. We keep saying it's not just a passive liner, it's a dynamic organ.
Absolutely. You can argue it's one of the most metabolically active and functionally diverse cell types in the entire body. Its roles go way beyond just containing the blood. Think about its absolutely critical role in hemostasis blood clotting.
It has to manage clotting right. Prevent it usually, but trigger it when needed.
Exactly that paradox. In a healthy, intact vessel, the endothelium actively works against clotting. It secretes things like prostacyclin and nitric oxide to inhibit platelet aggregation, and molecules like thrombomodulin to interfere with the coagulation cascade.
Keeping things flowing smoothly.
Right. It also releases tissue plasminogen activator, TPA, which helps break down any small clots that might form. But if the vessel gets injured,
Then it flips the switch.
completely flips. The underlying matrix is exposed, and the endothelium itself releases key pro-clotting factors. A huge one is von Willebrand factor, VWF.
VWF involved in platelet sticking.
Essential for it. VWF acts like molecular Velcro, helping platelets adhere to the site of injury and to each other, forming the initial platelet plug. So the endothelium is constantly balancing these pro- and anti-clotting signals.
And it's involved in hormone processing too.
Yes. A major role there is converting the inactive hormone angiotensin I into the very potent vasoconstrictor angiotensin II. This happens right on the endothelial cell surface via an enzyme called ACE.
ACE, the target of ACE inhibitors for blood pressure.
The very same. And endothelial cells also have specialized storage granules.
Storage granules. For what?
These are called Weibel-Palade bodies. They're like little intracellular packages containing pre-made molecules ready for rapid release. The two key contents are VWF, which we just mentioned for clotting, and P-selectin, an adhesion molecule crucial for inflammation.
P-selectin.
Yeah.
We'll get to that with inflammation, I bet.
You will. And don't forget, simple transport endothelial cells have lots of little vesicles, caveoli, involved in transcytosis, shuttling fluids and molecules across the cell layer.
Mechanically, they have to respond to the forces around them too, right? The blood flow.
Incredibly sensitive. They have specialized ion channels and receptors that detect both stretch from blood pressure and shear stress, the frictional drag of blood flowing over them.
And what do they do in response?
They react by releasing signaling molecules like nitric oxide, which causes smooth muscle relaxation and vasodilation. This helps regulate blood flow based on demand. And in the smallest resistance vessels, there's even direct communication.
Direct communication.
Yes.
Between endothelium and smooth muscle.
Yeah, through myoendothelial gap junctions. These are little tunnels directly connecting the endothelial cell cytoplasm with the underlying smooth muscle cell cytoplasm.
So they're electrically coupled.
In some cases, yes. It allows for really rapid coordinated responses between the lining of the vessel and the muscle controlling its diameter. Integrated function.
Okay. That integration must be absolutely critical during an immune response when white blood cells need to get out. Let's trace that leukocyte-endothelial interaction, the adhesion cascade.
Right. This is a beautifully orchestrated, multi-step process. It involves three main families of adhesion molecules acting in sequence.
3 families.
Yep. Selectins, integrins, and members of the immunoglobulin superfamily like ICAM and VCAM.
Okay, step 1.
Step 1 is rolling.
Yeah.
When there's inflammation nearby, the endothelial cells get activated. They start expressing selectins on their surface. P-selectin comes out rapidly from those Weibel pallid bodies.
The prepackaged stuff.
Exactly. And E-selectin is synthesized a bit later. These selectins on the endothelium grab onto specific carbohydrate molecules on the surface of passing leukocytes, like neutrophils.
But it's a weak grab.
It's a relatively weak transient binding. It's strong enough to slow the leukocyte down from zooming past, but weak enough that the force of blood flow keeps breaking the bonds. So the leukocyte tumbles or rolls along the endothelial surface.
Rolling allows it to sample the environment.
Precisely. As it rolls, it's exposed to chemokines chemical signals released from the site of inflammation, which often stick to the endothelial surface.
And recognizing those chemokines is the trigger for the next step.
That's the key trigger. Chemokine binding activates signaling pathways inside the leukocyte. It's called inside-out signaling. This rapidly changes the shape of another set of adhesion molecules on the leukocyte surface, the integrins.
Integrins change shape.
They switch from a low-affinity, kind of floppy state to a high-affinity extended state. Think of it like switching on molecular superglue.
And what do these activated integrins stick to?
They bind tightly to their partners on the endothelial cell surface. Molecules like ICAM1, intracellular adhesion molecule 1, and VSAM1, vascular cell adhesion molecule 1, which belong to that immunoglobulin superfamily.
And that binding is strong.
Very strong. This high-affinity integrin binding leads to firm arrest. The leukocyte stops rolling and flattens out against the endothelium.
Okay, stop. Now it needs to get through the wall. Diapedesis.
Right, transmigration. The arrested leukocyte then actively squeezes its way through the endothelial layer. This happens primarily in those postcapillary venules we mentioned because their junctions are looser.
Does it go between the cells or through them?
It can do both. Paracellular migration is squeezing between adjacent endothelial cells. Transcellular migration is actually tunneling right through the body of an endothelial cell.
Wow, through the cell.
Yeah, less common, but it happens. And inflammatory mediators released locally help this process by temporarily disrupting the endothelial cell junctions, making them even leakier.
And that leakiness contributes to swelling.
That's a major cause of the edema, the swelling and redness you see with inflammation, increased vascular permeability.
Now, you mentioned lymphoid tissues have special venules for massive traffic.
Ah, yes. The high endothelial venules, or HEVs, these are unique. You find them in lymph nodes, Peyer's patches, tonsils, basically all organized lymphoid tissues except the spleen.
What's different about them?
Their endothelial cells are not the usual flat, squamous type. They're plump, cuboidal-looking cells. They're metabolically very active and have specialized surface molecules for lymphocyte binding.
Designed for lymphocyte homing.
Exactly. Their cell junctions are also somewhat discontinuous, specifically adapted to allow enormous numbers of lymphocytes circulating in the blood to constantly exit and enter the lymphoid tissues for immune surveillance. It's a massive, ongoing migration process.
Okay, shifting gears slightly. We touched on shunts earlier, bypassing capillaries.
Yeah.
Can we elaborate on how the body does this, especially for controlling temperature?
Right. Arteriovenous shunts, or anastomoses, are direct connections between the arterial and venous sides, completely bypassing the capillary bed.
Are there different kinds?
Yeah, you can think about it at different scales. In the microcirculation itself, sometimes you have these metarterials, also called thoroughfare channels. They offer a slightly more direct path from a terminal arteriole to a post-capillary venule, allowing some blood to bypass the true capillaries within that bed. You see this in places like the mesentery.
Okay, small bypasses. What about larger scale shunts for things like temperature?
Those are the true arteriovenous anastomoses, AVAs.
Hmm.
These are more substantial, direct connections. Often they have really thick muscular walls, much thicker than the parent arteriole or venule.
Thick walls for control.
Absolutely. They are under strong control from the sympathetic nervous system. Prime examples are found in the skin, particularly in areas involved in heat loss. The fingertips, toes, ears, nose, lips.
How do they work for thermoregulation?
These AVAs often form specialized structures called glomera, singular, glum vomis. When your body needs to lose heat, sympathetic nerve input decreases, the thick muscle wall relaxes, the shunt opens wide, and large volumes of warm arterial blood flow directly into the superficial venous plexuses close to the skin surface, radiating heat away.
And when you're cold?
Sympathetic activity increases, the muscle constricts powerfully, the shunt closes down, and blood is forced through the deeper capillary beds, conserving core body heat. It's a very effective mechanism.
Fascinating. And related to blood supply patterns, there's this concept crucial for surgeons, angiosomes.
Yes, the angiosome concept. It's fundamental for plastic and reconstructive surgery. An angiosome is basically a three-dimensional block of tissue, skin, muscle, bone, everything that is supplied by a single-named source artery and drained by its accompanying veins.
A defined territory of supply.
Exactly. The source artery gives off smaller perforator vessels that supply that specific block. Surgeons need to know these territories, these angiosomes, or corresponding venosomes for drainage, when they're designing tissue flaps or grafts.
To ensure the flap survives.
Precisely. They also need to understand how adjacent angiosomes are connected. Sometimes they're linked by true anastomoses, where vessels of similar size connect. Other times it's via choke anastomoses, where the connecting vessels are much smaller, providing less reliable collateral flow if the main source artery is compromised. Planning surgery around these angiosome maps is critical for success.
Okay, we have the full, complex architecture laid out. Now let's confront the major diseases that target this system, starting with the big one, atherosclerosis.
Atherosclerosis. Yeah, arguably the most important and impactful arterial disease. Fundamentally, it's a disease of the intima, that inner layer. It's considered a chronic inflammatory process.
Triggered by injury.
Triggered by chronic or repeated injury to the endothelium. Major culprits are things like high blood pressure, smoking, diabetes, and especially high levels of LDL cholesterol, the bad cholesterol.
What does the LDL do?
LDL particles can get trapped in the intima. There, some of them become chemically modified, particularly oxidized. This oxidized LDL is a key inflammatory trigger.
Inflammatory trigger, meaning it attracts immune cells.
Exactly. It causes the endothelium to express adhesion molecules, attracting monocytes, a type of white blood cell from the blood. These monocytes squeeze into the intima.
And then what happens to them?
Inside the intima, they differentiate into macrophages and start gobbling up the oxidized LDL using special scavenger receptors. As they gorge on this lipid, they transform into characteristic bloated foam cells.
Foam cells. Sounds descriptive.
They look bubbly under the microscope. An accumulation of these foam cells forms the earliest visible lesion of atherosclerosis, called the fatty streak.
Is the fatty streak the dangerous part?
Not usually by itself. The real problem comes later. The chronic inflammation continues. Smooth muscle cells from the underlying media are induced to proliferate and, importantly, to migrate up into the intima.
Smooth muscle moving into the intima. It doesn't belong there.
It doesn't. And once there, these smooth muscle cells switch phenotype. They start synthesizing large amounts of extracellular matrix collagen, mostly.
Using that matrix synthesizing ability we talked about.
Precisely. This deposition of matrix, along with continued lipid accumulation and cell debris, builds up a complex structure called a fibrofatty plaque, or atheroma.
And the plaque causes the problems.
Yes. The plaque thickens the intima, narrows the vessel lumen stenosis, restricting blood flow. This can cause angina, chest pain, or claudication, leg pain on exertion. More dangerously, the plaque can become unstable, rupture, and trigger sudden blood clot formation, thrombosis, right on top of it.
Leading to a heart attack or stroke.
Exactly. That sudden blockage of a coronary or cerebral artery is what causes myocardial infarction or ischemic stroke, devastating consequences.
Now, people often use atherosclerosis and arteriosclerosis interchangeably. Are they the same?
They're related but distinct. Arteriosclerosis is a more general term meaning hardening of the arteries. Atherosclerosis is one specific, very important type of arteriosclerosis, characterized by those fatty plaques in the intima.
So what else counts as arteriosclerosis?
The other main form, sometimes called arteriosclerosis because it affects smaller arteries and arterioles, involves thickening and hardening of the vessel wall without the prominent lipid deposition. It's more about changes in the smooth muscle and matrix.
Like what kind of changes?
You might see replacement of smooth muscle cells with collagen, making the vessel stiff. Or you can get hyaline change, where proteins deposit in the wall. The functional consequence is reduced arterial compliance. The vessels become stiff pipes instead of elastic tubes.
And that contributes to high blood pressure.
It's a major contributor, especially to age-related isolated systolic hypertension, where the top blood pressure number goes up because the stiff arteries can't buffer the pressure pulse effectively anymore.
Okay, one more specific pathology, looping back to smooth muscle control. Raynaud's phenomenon, that extreme cold sensitivity. What's the cellular mechanism?
Raynaud's is fascinating. It's an exaggerated vasoconstrictor response in the fingers and toes, sometimes ears and nose too, triggered by cold or emotional stress. The digits can turn white, then blue, then red as blood flow cuts off and then returns.
What causes that extreme constriction?
Well, normal baseline tone in these skin vessels is maintained by sympathetic nerves releasing noradrenaline, acting mainly on alpha-adrenergic receptors on the smooth muscle.
Okay.
But in people with Raynaud's, something unique happens when the local tissue gets cold. It involves a specific subtype of alpha-2 receptor, the alpha-2C adrenergic receptor.
Alpha-2C?
Yeah. Under normal warm conditions, most of these alpha-2C receptors are actually hidden inside the smooth muscle cell, stored near the ER and Golgi apparatus, not on the surface membrane.
Hidden away. Why?
Seems to be a way to keep sensitivity in check, but when the local temperature drops...
They come out.
Exactly. The cold triggers a signaling process that involves the cell's cytoskeleton, the internal scaffolding. This process actively translocates those stored alpha-2C receptors from inside the cell out to the plasma membrane.
So cold exposure dramatically increases the number of these specific receptors on the cell surface.
Dramatically. And since these receptors also respond to noradrenaline, suddenly having way more of them on the surface makes the smooth muscle cell hyper-responsive to the normal sympathetic signals. Result: severe, prolonged vasoconstriction.
That's a really specific molecular mechanism. Does it explain why it's more common in women?
It seems to. Research suggests that estrogen can potentiate this process, possibly by increasing the overall expression levels of the alpha-2C receptor. This might explain why Raynaud's is significantly more prevalent and often starts earlier in premenopausal women.
Okay, we've covered the intricate control by smooth muscle and the complex vessel architecture. Let's finally focus squarely on the central engine. Cardiac muscle, the powerhouse pump.
Structurally unique, right?
Very unique. It is cross-striated, like skeletal muscle, which tells you the sarcomeres are highly organized for powerful contraction.
But the cells themselves look different.
Oh yeah, they aren't long, unbranched fibers like skeletal muscle. Cardiac muscle cells, or cardiomyocytes, are shorter, typically around 120 micrometers long, and crucially, they are branched. They connect end-to-end and side-to-side, forming this complex, interwoven, anastomosing network.
And the nuclei.
Usually just one, sometimes two, large nuclei located right in the center of the cell, not pushed to the periphery.
Now, this muscle works non-stop for a lifetime. The energy demand must be absolutely enormous.
Astronomical is the word. The blood supply is incredibly dense. No cardiomyocyte is ever more than about 8 micrometers away from a capillary. That's how vital continuous oxygen delivery is.
And inside the cell, does that reflect the energy need?
Massively. If you look at the volume of a cardiomyocyte, about 35% of it is occupied by mitochondria.
A third of the cell is just power plants.
Pretty much. It reflects its absolute dependence on continuous aerobic oxidative metabolism to generate the vast amounts of ATP needed for constant contraction and relaxation.
Is there variation within the heart? Are atrial cells different from ventricular cells?
Yes, there are differences. Atrial cells are generally a bit smaller than ventricular cells. And a key feature of atrial cells, especially in the right atrium, is the presence of numerous small, dense granules in their cytoplasm.
Granules containing what?
They contain the precursor hormone for atrial natriuretic peptide, or ANP.
ANP. That relates to blood pressure.
Exactly. When the atrial walls get stretched, say by high blood volume or pressure, these granules release ANP into the bloodstream. ANP then acts primarily on the kidneys, telling them to excrete more sodium and water.
Which lowers blood volume and pressure.
Precisely. It's one of the heart's intrinsic mechanisms for regulating fluid balance and load.
Okay, so the cells are branched and interconnected. What physically and electrically links them together? You mentioned intercalated discs.
The intercalated discs are the specialized junctional complexes found only in cardiac muscle, connecting the cells end to end. They look like dense, dark lines stepping across the fiber under the microscope.
And they have different components.
They do. They're complex structures with parts serving different functions. Running across the fiber, in the transverse portions perpendicular to the direction of pull, you have strong mechanical junctions.
Like what?
Primarily desmosomes, which act like spot welds providing strong adhesion, and fascia adherens. The fascia adherens is really important. It's where the actin filaments from the very last sarcomere of the cell anchor. It's functionally similar to a Z-disc, transmitting the contractile force directly from the myofibrils of one cell to the next cell in line.
So the transverse parts handle the pulling force. What about the electrical connection?
That happens mainly in the lateral portions of the intercalated disc, the parts that run parallel to the length of the muscle fibers. These areas are packed with gap junctions.
The communication tunnels.
Exactly. These gap junctions provide low-resistance pathways for ions to flow directly from the cytoplasm of one cell into the next. This allows the electrical wave of excitation, the action potential, to spread incredibly rapidly throughout the heart muscle.
Making the whole heart contract as one unit.
Essentially, yes. It allows the atria and then the ventricles to function as an electrical and mechanical syncytium, a single coordinated unit, even though it's made of individual cells. Essential for efficient pumping.
All right, let's get into the cardiac excitation-contraction coupling, ECC. How does the electrical signal trigger the mechanical contraction here? It's different again, right?
It is fundamentally different from both smooth and skeletal muscle ECC. A key feature is the cardiac action potential itself. It's very long.
Long action potential.
Yeah, unlike the brief spike in nerve or skeletal muscle, the cardiac action potential has a prolonged plateau phase, lasting maybe 200, 300 milliseconds. This plateau is created mainly by an influx of calcium ions into the cell through voltage-gated L-type calcium channels.
Calcium comes in during the action potential itself. Is that the main trigger, calcium?
It's the initial trigger, but it's actually not enough calcium by itself to cause full contraction. This incoming calcium accounts for maybe only 25% of the total calcium needed.
Only 25%. Where does the rest come from?
It triggers the release of the rest from the internal calcium store, the sarcoplasmic reticulum, SR. This is the defining feature of cardiac ECC, calcium-induced calcium release, or CICR.
So the calcium entering from outside acts like a key to unlock a much bigger flood of calcium from inside.
That's a perfect analogy. That initial trickle of calcium through the L-type channels binds to specialized calcium release channels, ryanodine receptors, on the SR membrane. This binding triggers those channels to open wide, releasing a large puff of stored calcium, the other 75%, into the cytoplasm, which then activates the contractile proteins, troponin C in this case.
Is the plumbing, the SR structure, different here too?
It is. The SR in cardiomyocytes isn't quite as extensive as in fast skeletal muscle, and its relationship with the T-tubules, the invaginations of the cell membrane, is different.
How so?
The T-tubules in ventricles penetrate the cell at the level of the Z-discs of the sarcomeres. Where the T-tubule runs close to the SR, they form specialized junctions called diodes. It's typically one T-tubule alongside a single terminal cistern of the SR.
Diodes, not triads like in skeletal muscle.
Correct. Skeletal muscle has triads, one T-tubule flanked by two SR cisternae. This diode structure in cardiac muscle ensures that the triggering calcium entering via the T-tubule L-type channels is delivered very close to the SR release channels, making CICR efficient.
Okay, contraction achieved. How does the heart relax so quickly between beats? Got to get that calcium out fast.
Relaxation requires rapidly lowering the cytoplasmic calcium concentration. Two main players here. The most important for speed is the SERCA pump.
SERCA?
Sarcoplasmic endoplasmic reticulum calcium ATPase. It's a high-affinity pump located on the SR membrane that actively pumps calcium back into the SR using ATP. This quickly sequesters most of the calcium that was released.
But if you only pump it back into the store, won't the cell eventually get overloaded with calcium over time?
Good point. You need a way to get rid of the calcium that entered from outside during the action potential plateau. That's handled more slowly, mainly during diastole relaxation, by two mechanisms on the cell surface membrane, sarcolemma.
Which are?
The primary one is the sodium-calcium exchanger, NCX. It uses the energy stored in the sodium gradient, low sodium inside, high outside, to push one calcium ion out of the cell in exchange for bringing 3 sodium ions in. There's also a sarcolemmal calcium ATPase pump that directly pumps calcium out using ATP, but NCX is generally considered more important for extrusion.
This careful balance of calcium removal actually provides a way for the heart to adjust its force intrinsically, right? The staircase response?
Exactly. The staircase effect, or frequency-dependent entropy. Think about what happens if the heart rate increases.
Less time between beats.
Right. Less time during diastole. The NCX exchanger, which slowly removes calcium, has less time to operate between beats.
So less calcium gets kicked out.
Correct. A little bit more calcium remains inside the cell after each beat. This extra calcium gets pumped back into the SR by circa, leading to a gradual increase in the amount of calcium stored in the SR over several beats.
More calcium in the store means more release next time.
Precisely. More stored calcium means a bigger puff of CICR with the next action potential, leading to a stronger contraction. So, intrinsically, just by beating faster, the heart contracts more forcefully. It's a built-in regulatory mechanism.
And how does the nervous system, the sympathetic system, powerfully boost heart function, say, during exercise?
Sympathetic stimulation via noradrenaline binding to beta-1 adrenergic receptors on cardiomyocytes is the most potent way to enhance contractility, inotropy, and heart rate pronotropy.
What does beta-1 activation do inside the cell?
It activates a signaling cascade that increases intracellular cyclic AMP, CMP. CMP then activates protein kinase A, which phosphorylates several key targets involved in ECC.
Like what?
2 really important ones. It phosphorylates the L-type calcium channels, making them more likely to open and let more trigger calcium in. This boosts CICR and force development.
Okay, more force. Does it help relaxation too?
Crucially, yes. It also phosphorylates a protein called phospholamban, which normally inhibits the circa pump. Phosphorylating phospholamban relieves this inhibition, making circa work faster.
So it pumps calcium back into the SR more quickly.
Much more quickly. This leads to faster relaxation, lusotropy. So sympathetic stimulation cleverly increases both the force of contraction and the speed of relaxation. This is vital because at high heart rates, you need the heart to relax fully and quickly to allow enough time for filling before the next beat.
Finally, we have to address the heart's major limitation. Its inability to heal properly after serious injury, like a heart attack. Limited regeneration.
That's the tragic reality. Unlike skeletal muscle, which has satellite cells for repair, cardiomyocytes largely lose their ability to divide shortly after birth. They exit the cell cycle permanently.
So if cells die, they're just gone.
Pretty much. The heart can respond to increased workload, like from chronic high blood pressure or a leaky valve. By undergoing hypertrophy, the existing cells get bigger.
But bigger isn't always better.
Often it's not. This pathological hypertrophy is frequently associated with detrimental changes. The cells can reactivate fetal gene programs, switching to expressing less efficient protein isoforms, like favoring the slower beta-myosin heavy chain over the faster alpha-myosin heavy chain in the ventricle. The whole structure can remodel in ways that ultimately impair function.
And this maladaptive remodeling contributes directly to heart failure.
Absolutely. Heart failure is often characterized by this combination of energy deficit, the overworked heart can't keep up metabolically, and problems with calcium handling, often calcium overload, because circa isn't working well, and NCX might be trying to compensate ineffectively.
So treatments often target these pathways.
Yes. For a long time, drugs like digitalis were used, which inhibit the sodium pump, Na plus K plus ATPase. This indirectly reduces calcium extrusion via NCX, leading to higher intracellular calcium and stronger contraction, but it's a dangerous balancing act. Modern chronic heart failure therapy focuses more on suppressing those harmful remodeling pathways using drugs like beta blockers, counterintuitively blocking the sympathetic drive, ACE inhibitors, and aldosterone antagonists to try and protect the heart long term.
Is there any regeneration potential at all?
The dogma for decades was zero. More recently, there's evidence suggesting a very, very slow rate of cardiomyocyte turnover in adults, maybe around 0.4 to 1% per year. But this is far, far too slow to allow for any meaningful repair after a significant injury like a major heart attack. Scar tissue forms instead.
Okay, we've dissected the pump, the pipes, the control systems. We need to cover one last crucial parallel system, the network responsible for fluid balance and immune traffic, the lymphatic vessels.
Right. The lymphatic system often gets overlooked, but it's absolutely essential. Its main job is to collect all the excess fluid that inevitably leaks out of the blood capillaries into the interstitial space, the tissue fluid.
How much fluid are we talking about?
It's estimated to be around 8 liters per day. All that fluid derived from blood plasma filtration plus any leaked proteins has to be collected and returned to the bloodstream. That collected fluid is called lymph.
8 liters a day. Wow. And it's also an immune highway.
Primary highway for immune cells, yes. Lymphocytes constantly circulate through it, and it's the route by which antigen-presenting cells travel from tissues to lymph nodes to initiate adaptive immune responses. It also transports absorbed dietary fats from the intestine.
Structurally, how does a system start out in the tissues? Where does it begin?
It begins as blind-ended capillaries, or lymphatic capillaries. Unlike blood capillaries that form loops, these just start as little closed-ended tubes scattered throughout the connective tissue.
Blind-ended tubes. What are their walls like?
They're made of a single layer of endothelial cells, but they have unique features. They generally lack a continuous, complete basal lamina underneath the endothelium, and they don't have parasites wrapped around them like blood capillaries do.
Designed for easy entry.
Exactly. They're designed to be extremely permeable. The junctions between the endothelial cells are discontinuous, and there can even be large gaps, allowing fluid, large proteins, cell debris, even microorganisms to enter the lymphatic capillary easily.
How do they stop collapsing under tissue pressure?
They have special anchoring filaments. These are fine strands of connective tissue that extend from the outer surface of the lymphatic endothelial cells and attach to the surrounding collagen fibers in the tissue. When the tissue swells with excess fluid, these filaments pull outwards on the lymphatic walls, actually helping to hold the lumen open so fluid can drain in. Clever design.
So fluid gets into these blind-ended tubes, but there's no central pump like the heart for lymph. What makes it flow?
Lymph flow is much slower and lower pressure than blood flow. It's propelled by a combination of factors. Intrinsically, the larger collecting lymphatic vessels actually have smooth muscle in their walls.
Lymphatics have smooth muscle.
The larger ones do, yes. This muscle contracts rhythmically, squeezing the lymph along segment by segment. That's the intrinsic pump.
And extrinsically.
Flow is significantly aided by external forces. The contraction of adjacent skeletal muscles massages the lymphatics, pushing lymph forward, that's the muscle pump effect, similar to what helps venous return. Also, the pulsations of nearby arteries can compress lymphatics and help propel the lymph. Breathing movements also play a role.
Okay, so it's squeezed along. What stops it from flowing backwards?
Valves. Lymphatic vessels, especially the collecting vessels, are packed with internal valves. They are typically bicuspid, semilunar valves, very similar in structure to those in veins, but they are much, much more numerous in lymphatics.
More valve than in veins.
Far more. These abundant valves ensure that lymph flow is strictly unidirectional, always moving away from the tissues and towards the large collecting ducts in the chest.
And they give lymphatics a certain look.
Yeah, when a collecting lymphatic is full of lymph, the segments between the valves bulge out, giving it a characteristic beaded or varicose appearance. These valves are crucial for preventing backflow and pooling.
And if this drainage system fails?
If lymphatic drainage is insufficient, maybe due to surgery damaging vessels, like after breast cancer surgery, infection, congenital problems, or blockage by parasites, lymph fluid accumulates in the tissues. This leads to chronic swelling known as lymphedema. So, wrapping this all together, this deep dive has really let us synthesize the incredible complexity of the body's flow architecture. We've hopefully achieved a real mastery over several key areas.
Yeah, definitely.
The dynamic, incredibly plastic nature of smooth muscle, its diverse roles, and the sophisticated mechanisms controlling vessel diameter and organ function. That's foundational.
Absolutely.
Yeah.
And then the layered, functionally distinct architecture of the vascular tree itself, from the elastic shock absorbers near the heart, through the muscular distributors, down to the critical resistance arterioles and the varied exchange capillaries.
Right. And finally, the heart itself. Understanding the critical, tightly regulated mechanics of the cardiac pump, especially that unique calcium-induced calcium release mechanism and its vulnerability in disease.
And it feels like a recurring theme, especially with smooth muscle, but also relevant elsewhere, is this idea of phenotypic plasticity.
It really is. The ability of smooth muscle cells in particular to not just contract, but to also synthesize their own matrix, to proliferate, to migrate essentially, to change their job description throughout life.
It gives them amazing regulatory power.
But it also makes them central culprits in so many chronic diseases. That same plasticity drives the pathological remodeling we see in atherosclerosis, in hypertension, in the airway narrowing of chronic asthma.
So thinking at that professional level, where does this understanding lead us?
Well, considering this remarkable lifelong ability of smooth muscle, and we should probably include related cells like fibroblasts and parasites too, to switch their phenotype between contractile, synthetic, proliferative states. It raises a provocative question for the future.
Okay.
If pathological remodeling is fundamentally driven by this cellular plasticity going awry, could we potentially leverage those same capabilities? Could we use targeted therapies, maybe gene therapy or specific drug delivery, to intentionally manipulate the phenotype of these cells?
To reprogram them, essentially.
Maybe. Could we push them away from harmful synthetic or proliferative states back towards a healthy contractile state? Can we move beyond just managing the symptoms of diseases like hypertension or atherosclerosis and actually achieve a genuine cellular level reversal by directly intervening in smooth muscle cell behavior and its interaction with the matrix?
A huge challenge, but one that requires exactly this kind of deep foundational mastery of the underlying biology we've explored today.
Precisely. Something to think about.
Indeed. That concludes this deep dive into the architecture of flow.